My name's Dallin Bailey. I'm a group leader at the Pirbright Institute. I work on viral glycoproteins. So I'm a recent coronavirus biologist. So I've been working on coronaviruses since the start of 2020, but I'm standing in as co-director on this network for Erica Bickerton, who's been working on coronaviruses for a lot longer. She works a lot on IBV. Erica is on maternity leave. So today I'll just be introducing this countermeasures and interventions section, and we've got 4 talks, 2 this morning and 2 this afternoon, that should go into these areas in a little bit more detail. But I just thought I would frame the debate quickly in terms of what we mean by countermeasures and interventions. So as a molecular biologist, I think I certainly tend to think of countermeasures as vaccines and interventions as antivirals. But I think, the realization from the last 2 years has been that these two features of countermeasures and interventions must always be run in parallel with non-pharmaceutical interventions. And whilst I won't speak about it today in much detail, I just wanted to stress that these things really have to run together. So obviously, we're all very familiar of the success of the COVID-19 vaccination campaign, and I'll just perhaps briefly give a sort of historical overview of that from my own perspective in the next couple of slides. But I did want to stress, especially because this is more of a broader coronavirus network, that clearly the coronavirus vaccination didn't begin with COVID-19 vaccines, and that there are actually approved animal coronavirus vaccines to canine coronavirus, feline coronavirus, bovine coronavirus, 2 different porcine coronaviruses, and IBV. These are all licensed and in use. So, and that I think is something what the first thing I wanted to stress about the successful COVID vaccine development was really that this was building on state of the art. This didn't just arise out of the ether. There was a considerable base of existing knowledge that allowed the rapid development of the COVID-19 vaccine. And I actually pulled this slide from a review very early in 2020, where they were discussing the possible pipeline for rollout of vaccines. And I think what they were stressing here was the fact that MERS and SARS-CoV-1, there had been no vaccine for 6 and 17 years respectively since their emergence, and that even with this existing knowledge, it might take 12 to 18 months to really get to that position. And as we know, it obviously happened even quicker than that. But I think in terms of what led to this success, I think this is obviously integral, building on state of the art. So, all the coronavirus vaccine research that had been done before. And then clearly the capacity to have a broad spectrum of available platforms. So obviously in the UK, most of the success so far has been based on viral vectored vaccines. So the AstraZeneca vaccine from University of Oxford, and then the 2 mRNA-based vaccines from Moderna and Pfizer. But there is a huge selection of other platforms and vaccines going through clinical trials. And the other thing that sort of again stresses the importance of building on state of the art was that I think perhaps we were slightly fortunate with regards to SARS-CoV-2, that we had a clear identifiable target, and that being spike. And building on, again, state of the art, for instance, the double proline mutant to stabilize the spike in a pre-fusion form was incredibly important, although it's not in all of the vaccines. And you can see, there was obviously an urgent need, but this was met with an urgent provision. And this is the data from the New York Times data tracker showing the different vaccines and different stages of clinical trial here. And I've just summarized that there. This is actually from a different tracker, but 90 in development and 24 approved for use in different statuses. This was obviously supported by huge global vaccine initiatives like Operation Warp Speed and COVAX. but I think we're still left with the situation that there are ongoing issues. I think clearly the greatest concern being wider accessibility. And so this is a share of population receiving at least one dose. And clearly there are some continents that are lagging behind and need more assistance in able to vaccinate their populations. We're moving ahead with the booster campaign in the UK, but obviously we're concerned about variants, and so all of these things need to be continually monitored in the future. So in terms of what we've done at Pirbright to work to help with the COVID-19 vaccine research, so my lab works on viral entry. So we have for a long time been using pseudotype systems to look at entry and neutralization. And we had some long-standing collaborations with University of Oxford teams that were working on the Cladox vectored spike. So they, at the beginning of the pandemic, had performed or started a large number of immunogenicity and safety trials in different animal species, and we were running the VNTs for those in non-human primates and ferrets, and subsequently some studies in mice. And actually Pirbright led the safety and immunogenicity study in pigs, which are a very good model for human vaccines. And then more recently, again building on the pseudotype technology, we're part of the Genotype to Phenotype Consortium, and we're working with the UK Health Security Agency on a cohort of elderly patients, looking at their sera. And really what we're focused on is the emerging variants. So this is the beta variant, the mutations in spike, and we're trying to understand whether or not the variants can escape from the immune response generated by vaccine or infection, because that clearly is one of the most important things to consider in the future. So in terms of antiviral and drug development, this is the same tracker. Clearly this takes a little bit longer, or has taken a little bit longer in terms of development, but obviously there was very good news yesterday on molnupiravir, and we'll hear about that later this afternoon. I just again thought I would very briefly run through these. So monoclonals, clearly these were some of the first to come on board, and that's again building on state of the art. So the one from Regeneron and others have come on board. That was sort of supported by convalescent plasma. People have looked at recombinant ACE2 as a receptor decoy. We're now seeing more antivirals emerging, remdesivir, molnupiravir, favipiravir, coming on board, or in the case of remdesivir, perhaps being used less widely now. And then I think it's also been maybe not direct acting antivirals, but trying to deal with the inflammation. That's a big problem in COVID. There's been obviously a large number of studies looking at reducing that, and there are a number of drugs being licensed or tested in clinical trials, and or the old famous inhaled interferon. And then there's clearly been a number that have been questionable or failed, including the hydroxychloroquine and chloroquine issue. So just the last slide here for me, just in terms of what we're doing in terms of COVID-19 antiviral research. So like I said, my lab's interested in viral entry, and we developed in 2020 this quantifiable fusion assay for SARS-CoV-2. So the spike protein will confuse cells, and we use a recombinant GFP and luciferase construct, and then we can add antibodies to the effector cells to see if we can inhibit SARS-2 fusion. And the jury's a little bit out now on how much syncytia SARS-2 can form in patients, but we thought it would be interesting to set up an assay to quantify the ability of antibodies to inhibit this. And this is plasma from convalescent individuals showing a specific inhibition of that fusion. And we've now adapted that assay into a more robust, higher throughput format in it to allow us to screen the FDA drug-approved library of about 3,000 compounds, and we're churning through that work and doing some validation at the moment. So hopefully I've given you a sort of a taste of some of my thoughts on coronavirus vaccines and antivirals, and some of the work that we've been involved with.